You're listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is July 27, 2012, and I'm your host, Dr. Jay Portnoy. Our topic today, allergen extracts and standardization. Our presenter is Dr. Robert Esch. He's the Director of Research at Greer Laboratories in Lenoir, North Carolina.
I just wanted to disclose that I am an employee of Greer Laboratories, which is a provider of allergen products to practicing allergists in the U.S. And today what I wanted to do was give an introduction to allergen extracts, specifically to help you understand how allergen extracts are manufactured and controlled in the U.S., and to identify and compare the approaches used to standardize allergenic extracts. As you probably know, the majority of commercially available extracts in the U.S. are non-standardized, and so maybe we should be able to discuss what the differences are between standardized and non-standardized allergenic extracts. Allergen extract companies, there are 6 now. When I entered the industry, there were about 15 different allergen extract manufacturers. Many of them produced allergens that were indigenous to a given geographical area, but for various reasons they've consolidated, and there are 6 fairly large companies that manufacture all of the allergen products used in the U.S. That have been derived from pollens, epithelia, insects, mites, foods, for both diagnostic and therapeutic use.
You know, Bob, actually, I was thinking back to when we first started, and I remember that allergists used to make their own extracts, too.
That's true. That? Yeah, the methods employed by the manufacturers haven't changed since the early 1900s, when Roger Wodehouse, Arthur Coca, and Robert Cook developed the methods to make aqueous extracts for skin testing and immunotherapy. So much of the techniques that are used have been derived from these early pioneers who taught their fellows and their training programs how to manufacture them. And when they went out to their private practices, they would purchase the raw materials from companies like Greer, and they manufactured their own extracts in their offices. It wasn't until the 1940s and 50s when sterility became an issue with these homemade extracts that the FDA decided to become involved in the regulation of these extracts, and that's when more and more of the companies took responsibility for manufacturing the bulk extracts. But it might be also important to point out that interest in how these extracts are manufactured and standardized should be of interest to practicing allergists, because in the U.S. these extracts are still formulated in many of the allergy practices to fulfill named patient therapeutic mixtures in the offices. So it's not totally, if you include that as part of manufacturing, there is a degree of manufacturing that still occurs in the practicing allergist clinic. So as I pointed out, you know, hundreds of extracts are manufactured from pollens, fungi, epithelia, food, and a variety of insects. The last count was about 1,200 extracts were recently reviewed by the advisory board to FDA in a recent reclassification of non-standardized extracts. Now, most of the pollens and animal-derived source materials and foods are natural products that are purchased, the source materials are purchased from collectors, which collect the pollen in their natural state, and probably only the fungi and many of the arthropods or insects and mites are cultured by the manufacturers for their source materials. Now, there are a variety of formulations. Once the extracts are made from these source materials, the final products that are provided by the manufacturers include aqueous formulations, which are usually saline or buffered saline formulations, including 0.4% phenol as a preservative. Since these extracts are in multiple dose containers, there's an FDA regulation that requires some sort of preservative to be added. And 0.4% phenol is the most commonly used preservative in allergen products. Now, glycerinated or 50% glycerinated extracts have an added advantage of increased stability and also serves as a preservative, as an antimicrobial preservative. So some 50% glycerinated extracts will not contain phenol, because as long as you maintain these formulations in greater than 50% glycerin, it has the antimicrobial effectiveness equal to that of added phenol. Now, this glycerinated formulation has some popularity among certain clinicians that believe that phenol can be a source of allergies. So there are physicians without proof that it is IgE mediated that they want phenol-free formulations, and it's only the glycerinated products that can provide this. Now, there were lyophilized and alum precipitated extracts, but the added benefit of increased shelf life of the lyophilized preparations really didn't take hold, because most allergists wouldn't store extracts for years and years. They used the products relatively quickly, and so the lyophilized extracts have basically disappeared from the clinician's office. Now, these different formulations are used for diagnostic and therapeutic use. Now, the prick puncture or diagnostic skin test materials are provided in glycerinated form, because that's the most common formulation used for prick testing. For intradermal tests, dilutions are made in aqueous saline buffer, because glycerin can be an irritant when delivered intradermally. So it's important to use aqueous formulations for that purpose. And then for therapy, a variety of aqueous and glycerinated preparations are usually employed. Now, the pollen source materials, as I said earlier, are collected in nature, either by sieving, vacuum collecting, or water setting in pollen sheds. Now, it's important that when pollen is collected by sieving or vacuum where the plants are grown, they have to be monocultures, and care has to be taken that no other aeroallergenic plants are pollinating at the same time. So the surest way to maintain purity of pollen that's collected is to water set them in specific pollen sheds indoors. But with ragweed, as you can see in panel B, about 15 years ago, or 20 years ago, that NIH was collecting ragweed pollen for purifying MV1 and the other ragweed allergens. We had a contract to collect 50 kilos of ragweed pollen within a year, And so we planted ragweed and devised a vacuum collection device that we put onto a tractor and just drove it through the ragweed fields and collected 30 kilos in one season in one field.
I bet the neighbors were just thrilled.
Now, fungal source materials are cultivated in the laboratory. But to give you an example of how complex this can be, is that even for a single fungal allergen like Alternaria alternata, since we know that over 100 different strains exist through ATCC, that each one of these strains could give a different allergen profile or antigenic profile, and so the selection of the appropriate fungal source materials used for extract manufacturing can be a very complex decision, and also the media that's used to grow can lead to different antigenic profiles within the same strain. And to add further complexity, whether a company utilizes the mycelia, the spores, or the culture filtrates, antigens in their extract can lead to this increased variability. So even though we have the controls of laboratory cultivation, the fungal extracts are probably the most variable of all allergen extracts that are available. Now, in terms of regulations, the identity and purity of pollen source materials or any allergen source material is probably the most important quality control criteria, especially for materials that are collected from nature. So with pollens, usually a confirmation of the genus and species through botanical specimens, and confirmation by looking through the microscope at the pollen grains, because at least each genera or group of plants produce a fairly identifiable characteristic pollen grain. Now there are some pollen grains, such as among the grass pollens, that even experts can't distinguish the two. Then that's when we would have to resort to specific allergen determinations, such as low P1 or group 1 allergens of grass pollens, to differentiate different species of pollens. But for the most part, microscopic identification and identity confirmation by looking at plant materials is the method used. With fungal source materials, as I was saying, that even within Alternaria, a single species, different strains show different characteristics depending on the growth media used. So we use certain growth profiles, such as in the panel, the upper left panel, you see SAB, that's Sabarod's media. PDA stands for potato dextrose agar. And you see 3 different strains, A, B, and C, and their growth characteristics. So strain A will grow mainly in the mycelial form and does not produce pigmented spores in either media. Whereas in strain C, you would see when grown on potato dextrose agar, produces a high concentration of melanin, a dark pigment contained in the spores, but on Sabarod's media produces much less pigmented spores. So just sporulating characteristics tell us a lot about the strain and the identity for stability. So it's important that since fungi are highly mutable, that there is a consistency in terms of metabolic characteristics produced by strains. The lower panels show highly mutable strains. You can see the segmentation in the right side. which leads to a mutation by the alternaria to become an asporulating. The white sectors show growth of a non-sporulating mutation from divisions from a single spore that was inoculated in the center of the plate. As it grows out radially, it creates this segmenting pattern which shows the high mutation rates in certain strains of Alternaria. And these kinds of strains, by all means, are avoided for use in production of allergen products. Now, in terms of the typical extraction and quality control steps used, I mean, we start with the allergen source materials where the identity and purity is confirmed, and then extractions are prepared in aqueous buffers to manufacture what we call bulk intermediates. Now, these bulk intermediates are fairly large volumes, usually on the order of 20 to 50 liters, where sterility, safety, and a variety of potency assays are conducted. So these are stock concentrates that can be provided directly to the allergist. So they're usually vialed in large bulk containers containing up to 50 mils of bulk intermediates provided as concentrates. Now these concentrates have to be further diluted or mixed or formulated into named patient products, either in our manufacturing facilities or in the allergist's office. Now normally when we do the formulating, we have to do additional sterility, safety, and chemical tests to confirm that the dilution or the mixtures were conducted according to either the prescription or the manufacturing procedure. Now, if it's conducted in the allergist's office, there are USP regulations that recently have been implemented, namely related to training of the technicians that formulate these extracts on aseptic technique, and also in terms of record keeping to be able to trace the composition of the mixtures.
Just for the fellows' information, here at Children's Mercy, we write prescriptions for extracts, and then they're prepared at Greer Laboratories. So Greer does the preparation of the patient-specific vials for our office. Most offices probably buy the stock bottles and manufacture their own. So,
yeah, usually the larger practices will have formulating labs within their practice, but most of the solo practices or smaller allergy practices usually have pharmacies associated with each of the allergen manufacturers to produce them. And this is one of the main differences between the practice of allergy in Europe and in the U.S. In Europe, all allergen named patient products have to be formulated by the manufacturers, whereas in the U.S. I would estimate that up to 90% of all named patient immunotherapy is formulated within the allergist clinic. Now, allergen standardization, and this is a practice that's important if we're to assure batch-to-batch consistency. I mean, before the advent of standardization, each lot of extract, you could expect as much as 10-fold variation in potency. So, I mean, that was considered to be consistent, and that was one of the reasons why each time an allergist obtained a fresh or a new lot from a manufacturer, they would reduce the injection up to 10-fold to ensure safety of to the patient who was receiving an immunotherapy injection. So safety is the main reason for the need of allergen standardization in order to assure batch-to-batch consistency, not only in the composition, but in terms of the relative potency of each lot. Also, the safety improvement in terms of effective doses. So not only from a safety point of view, we're beginning to understand what the minimal effective doses are for immunotherapy. And in order to ensure that each patient receives an effective dose, or at least a target effective dose, some sort of labeling of the potency of the extract should be available to the practicing allergists. And also, standardization is important in terms to give shelf life data or stability information about bulk intermediates as well as patient-specific therapeutic mixtures. So one of the main differences between standardized and non-standardized allergen extracts is that standardized extracts have a unit of potency that appears on the label, and each lot is tested for conformance to a U.S. standard. So at present, I've listed the standardized extracts that are available and how and what type of assays are performed. For instance, with the venoms, the Hymenoptera venoms, hyaluronidase and phospholipase, two important allergens in the venom. The activity is measured, and each of the, although on the label it's reported as total venom protein content, and the doses are targeted based on the protein content, hyaluronidase and phospholipase activity must be within a certain limit based on specific activity measurements. With ragweed pollen, it's measured by the major allergen content, MBA1, by radial immunodiffusion. House dust mites are tested using a ELISA inhibition assay with respect to relative potency to a U.S. standard, in much the same way that grass pollen allergens are standardized. And for cat hair and cat pelt, the major allergen FeLV1 content is measured for each lot. In comparison, non-standardized extracts have no unit of potency, and they're labeled based on the weight by volume extraction ratio, meaning that if it's labeled as 1 to 10 weight by volume, it refers not to the dissolved weight of solute in the volume of extract. It refers to the extraction ratio, meaning that 1 gram of source material was extracted in 10 milliliters of extraction fluid, and whatever material that was solubilized in that ratio is its content. So 1 to 10 weight by volume refers to the extraction ratio, and that's important to remember. Or PNU, which stands for protein nitrogen unit. It could be some extracts are labeled based on a Keldol nitrogen determination of the soluble extract. Now, neither one of these designations necessarily reflect the allergenic activity of the extract. So the thing to remember is that if we were to conduct stability studies, so the weight by volume by its definition will never change even after 100 years, so that type of labeling will never reflect or could never be used to ensure potency or shelf life. Okay. So how are expiration dates of non-standardized allergenic extracts determined? Well, if we had a potency test for standardized extracts, we do stability studies, and so the dating that you see on the vial reflects actual stability studies that each manufacturer has conducted. But with the non-standardized extracts, the shelf life or dating period is determined by law. Okay, so back in the 1930s, an advisory committee to the FDA of practicing allergists determined, or at least they knew that glycerinated extracts were more stable than aqueous extracts, but they arbitrarily gave 3 years of dating after a glycerinated extract leaves the manufacturer. So I guess they assumed that allergists didn't store extracts in a controlled manner. So they said, when we ship non-standardized extracts, we apply a shelf life of 3 years for glycerinated extracts.
Are you allowed to store it in the manufacturer plant?
3 years as well.
So it could be 6 years.
The maximum shelf life of a glycerinated extract, if we ship the extract the day that it expires in our hands, we could add 3 additional years. Well, it turns out that, we don't, because of manufacturing planning, most of our extracts, glycerinated extracts, leave our site within a year. Aqueous extracts are allowed to have 18 months of dating after it leaves, plus 18 months within our storage. So you can have up to 3 years total. And again, most allergists want very fresh aqueous extracts because the stability is not that great. And we try to at least ship within 12 months after manufacture. So it's quite arbitrary how, I think the thing to remember is that the dating and the shelf life of non-standardized extracts are not determined by potency testing or specific allergen tests. Now, how are allergen extracts standardized? Well, these biological equivalent units that you hear about in terms of labeling has to, or at least the FDA has deemed it, that the biological activity must be related in some way to the labeled potency. So merely measuring a specific allergen content in mass units is not sufficient. We have to be able to relate that value, especially for AMBA1 and FeLD1, where major allergen determinations are the laboratory test. Somehow or another, we have to refer that to a biological activity. And in the U.S., the ideal approach, which is the activity of allergen extracts when delivered intradermally into highly allergic patients. So I wanted to spend a few moments to go through the procedure by which reference preparations are assigned this bioequivalent allergy unit. This is specific to U.S. standardized products. So in the ideal method, at least 15 highly sensitive allergic subjects based on history and prick test response are selected. And I think it's real important that it's not to point out that these 15 subjects are selected to represent the most sensitive of that allergic population. So it's not representing what a normal, for instance, if it was ragweed that we were trying to standardize, that you wouldn't get a normal distribution. You want to skew these test subjects to represent the most sensitive within ragweed sensitized subjects. So with the selected subjects, threefold serial dilutions are prepared and delivered intradermally, and the erythema response, not the wheal, but the erythema response is measured and then plotted against the dilution, the number of threefold serial dilutions, and the sum of erythema. And if the dilution can be made 14 times, if 14 threefold serial dilutions can be made from the stock concentrate, okay, and gives a 50 millimeter, and that's where the ID 50 comes from, the 50 millimeter sum of erythema, that dilution, 14, represents 100,000 BAU per mil. Okay, so a extract labeled 100,000 BAU per mil, if you take a statistical sample of most highly allergic individuals to that extract, you should be able to dilute that extract 14 threefold, dilutions, and when you administer that intradermally, it should elicit a 50 millimeter sum of erythema response in those subjects. Okay? So a reference preparation that has been evaluated in these subjects is diluted exactly to 100,000 BAU. Now, that reference is now used in ELISA inhibition relative potency measurements, and that relative potency is what's used to assign the BAU for every subsequent lot of allergen extract that's manufactured.
So you don't have to round up the 15 sensitive people and retest them every time you make a new lot.
Exactly. So we can use an in vitro surrogate using a serum pool, an IgE serum pool from those characteristic allergic patients, and the reference preparation that has been skin tested is used as a reference dose-response curve, and every subsequent lot is compared to that reference, and when it becomes equivalent in terms of its inhibitory potency in the in vitro test, then we can normalize the potency of the bulk intermediate and adjust the potency to be a relative potency of 1.0, which we would assign to 100,000. Okay? Now, in terms of major allergen content, ragweed and cat extracts are labeled based on FeLD1 and AMBE1, respectively. Now, in order to deem an allergen as a major allergen, there are certain rules that the FDA would want to see fulfilled before a major allergen determination can be a surrogate for allergen potency. One is that a major allergen is an allergen that is responsible for more than 10% of the total IgE antibodies to the whole extract, and that the majority of patients that are allergic to that extract must be sensitized to that specific allergen. And secondly, the concentration of that major allergen must correlate with the overall biological activity as determined by ideal testing or IgE ELISA inhibitions. Now, with the FeLD1, the method by which FeLD1 determinations became a surrogate is shown in this slide, that when Paul Turkeltaub, then at the FDA, compared commercial extracts and determined the relative potency based on skin testing, which is shown in the y-axis, with the FeLD1 content of each of these extracts. And so you can see some extracts that had high FeLD1 content had correspondingly high skin test reactivity, And those extracts that had low FeLD1 content had proportionally low skin test activity. And so when you plotted this, you see a very strong relationship between the FeLD1 content of a cat extract, regardless of how it was made amongst the different manufacturers that were selling the material, with its overall. So this was enough evidence that the FeLD1 content of each of the manufacturer's extracts predicted the biological activity. And on this basis, FeLD1 became defined as a major allergen from a standardization point of view. Now, if you look at cat extracts, FeLD1 is not the only possible allergen. There's two types of standardized cat extracts. One, on the label, it would refer to it as cat hair, and the other is cat pelt. They both contain the same amount of FeLD1, targeted at 15 units of FeLD1 per mil for those extracts that are labeled 10,000 BAU per mil. But there's one major difference between the two kinds of extracts. They're labeled the same. If you look at the albumin, FeLD3 I believe it is, that cat pelt contains 10 to 20 times more cat albumin than cat hair extracts. Now this becomes important in those minority of cat allergic patients that are sensitized to albumin. For the majority of cat allergic patients, those that are sensitized only to FeLD1 or predominantly to FeLD1, the two different extracts would probably be interchangeable. But it's important to remember that there are cat allergic patients that will be sensitized to albumin, and so the selection of these two standardized products becomes important, not only in terms of its diagnostic efficiency, but also in success in immunotherapy.
Would there be a value to testing for both of them routinely then?
Some allergists do treat with both and test with both.
Wow. Because you can't just get cat albumin by itself.
That's right, you can't. That's right. Now, short ragweed extracts are available in, or for the most part, it's glycerinated extracts, but some manufacturers also provide aqueous extracts. And there is one manufacturer that labels short ragweed extract on the basis of AU per mil. But for the most part, they're labeled both, and since short ragweed extracts was the first standardized extract made available, most of the manufacturers still label them on the basis of weight by volume, but in addition to that, will label each vial with the MB1 content, which could be highly variable. So cat extracts, every lot of cat extract has approximately 15 units of FeLD1 per mil and is labeled 10,000 BAU per mil. But short ragweed extracts can be purchased with as little as 200 MB1 units to as much as 800 MB1 units. And so if you're dosing based on major allergen content, you would have to adjust your formulations. If you're targeting an MB1 content, you would have to make those adjustments based on the labeled value. Okay. Now, dust mite extracts are all labeled based on AU or BAU per mil. They're only available in glycerinated formulation. Now, between the two different species of mite, there is some variability in terms of its group 1 or DIR1 content, but for the most part, both the D. farini and D. ternicinus extracts contain roughly the same amount of DIR1. Now, as we know, DIR1 is not the only important allergen in mites. DIR2, or the group 2 allergens, are important. Now, although most manufacturers in the U.S., there is a strong correlation between group 1 content and biological potency. When the FDA compared the two using that, remember the skin test versus SPELD1 content that I showed you with the cat extracts, that relationship was not as strong with mite extracts. And that's the reason why DIR1 content alone is not the basis for standardizing mite extracts. Grass pollen extracts, between the 8 different grass pollens that are standardized, 7 derived from temperate grasses such as meadow fescue and timothy, and 1 subtropical species, namely Bermuda grass, are labeled either 100,000 BAU for the temperate grasses and 10,000 BAU for Bermuda grass. Now, if you look at the group 1 content and the group 5 content, the variability is even greater than what we saw with the group 1 mite allergens. And for this reason, the group 1 major allergen, if you define it as such, is not used because up to 10 other allergens have been identified and shown to be important depending on the patient examined. Now, one of the advantages of standardization, I think, could be illustrated by this slide. Before standardization, when grass pollen extracts were available only on the basis of weight by volume, if you compared the BAUs determined by skin testing for each of these 8 different allergens, you can see the potency range of commercially available extracts at the time. So if you look at Bermuda grass, 1 to 10 weight by volume extracts that were available prior to standardization, the potency ranged about 8-fold from 5 to 40,000 BAU per mil. If you go to meadow fescue, you see another close to a 10-fold difference between the most potent commercially available extract at the time, which approached 1 million BAUs per mil. That's 10 times higher than the most concentrated extract that you can purchase today. And I think in many ways, if when we looked at adverse reactions to immunotherapy before standardization, I would venture to guess that grass pollen extracts were one of the most frequently reported formulations that led to adverse events.
Well, we were overdosing everyone.
Yeah. So the doses, if you look at the targeted doses that are in the practice parameters today, and you look at how some allergists were practicing immunotherapy, that many patients were probably getting 10 times more allergen than they needed. I think we already covered the Hymenoptera venoms. This is the table, an abbreviated table from the practice parameters that show the effective target doses of each of the standardized extracts as well as for non-standardized extracts. And I think as standardization is implemented for the other non-standardized extracts, we'll be able to, this table will give us much more information that is needed to be able to determine effective doses and as well as safe doses.
I see this table all the time. Do you agree with this? Do you think that it's pretty good?
Well, in terms of, I mean, they are based on generic extracts. One of the problems with this table is that a lot of this information comes from different manufacturers' products. A lot of it from European products that are modified allergens. You know, So based on the overall activity, though, I think they serve as good target doses. We're finding that most allergists will try to achieve these doses, but in many cases they cannot. But we know that if you're within 5-fold, or even within 10-fold of these extracts, doses, that a lot of the clinical trials show that you're within the effective dose range.
Okay.
So they serve as good target doses for maintenance therapy.
They at least give us some guidance. Sure.
Exactly. And the dose adjustments usually have to be made based on patients. I think if each of the package enclosures will, they'll give you the kind of a dose target, but each patient is rarely given the same dose regimen. Now, one way to reduce the amount of allergens that you put in an extract, I think with the 1,200 different extracts that are made, I mean, we've always, and as you add more and more extracts, there's a dilution effect in immunotherapy mixtures, and if they're highly cross-reactive, you can actually skew the amount of allergen that you're giving depending on the number of cross-reacting allergens that you end up including. Now, examples of cross-reacting groups for non-standardized extracts, I think a good guide would be to use families or subfamilies. So there is a relationship between taxonomic groups and cross-reactivity. So what it's also in the practice, included in the practice parameters. Knowledge a little bit about plant taxonomy will go a long ways into selecting what allergen extracts to include in your diagnostic and therapeutic panels. So in summary, as I tried to illustrate, no single allergen standardization approach is suitable for all allergenic extracts. Where major allergens exist, they can serve as a surrogate for biological activity, and in those cases, they have been used. Now, the important aspect of standardization is it reduces the lot-to-lot variability in allergen potency, which will increase the safety and hopefully will eliminate the need to make dose adjustments each time you get a new manufacturer's stock concentrate, as it's been widely practiced in the past. And then finally, the importance of the allergen potency testing is that the manufacturers can rely on expiration date that is based on validated stability tests. And then the implementation of clinical practice parameters, namely in terms of targeting doses, is highly dependent on progress made in allergen standardization.
All right. Well, very good. Well, thank you so much for that presentation. I think that's, yeah, did you want to go ahead and take back the slideshow? So are there any comments or questions from the audience about Dr. Esch's presentation? I guess I would like to know are there any new extracts that are being considered for standardization? It's been quite a while since we've had any new ones that have become available. What's being worked on now?
Well, there's been two initiatives. One that was actually proposed by the FDA, and that is cockroach allergen extracts, mainly because of the interest in the Intercity Asthma Consortium and their interest in allergens that are important in asthma.
Right.
So cockroach is one. And the other among the manufacturers has proposed that dog epithelial or dog dander extracts be standardized.
Yeah, there's the issue of the CAN-F1. There's the AP dog and then the regular dog, and there's kind of a question of which one should we be using, and how do we How would you address that question?
Okay, what we're finding is that it is very similar to the situation with cat extracts in that the dog allergens, there are two important allergens, CanF1 and CanF3, which is dog albumin. But as compared to the cat allergic patient, the albumin seems to be much more important in a larger percentage of dog allergic subjects. So I think there is a need, like if you look at the AP dog, it's mainly CanF1.
Right.
With very little dog albumin. And then the dog epithelial extract has relatively low concentrations of CanF1 and high concentrations of CanF3 or albumin.
So maybe the combination of those two, depending on
combination of the two is probably will lead to the optimal extract. But in that case, both types of extracts I think are important in the clinic.
Okay, great. Any other questions from anyone?
Dr. Esch, we had a question. This is Anna Argo, one of the second-year fellows. Dr. Nelson gave us a lecture not too long ago on immunotherapy, and we were talking about the stability of the different allergens. And, what we typically do here is that we try to put all the pollens in one, and then we end up putting cat and dust mites with any of the molds. And he was saying that really mold and cockroach should not be together, nor should they be with anything, that they can even degrade dust mite and cat. We went back and looked at some of the different literature on this and found that maybe glycerin content affects us, and so we're wondering what you thought, since we use Greer here, in terms of what glycerination context we use and the protease activity of those.
Yeah, that's been quite a large interest in our laboratory here, is to look at stability of various combinations. And as you point out, the glycerin concentration in the final mixture is critical in determining the relative shelf life. The higher the glycerin content, the more stable any extract will be.
Right.
In terms of the cockroach, however, cockroach extracts have such high protease concentration that even at 50% glycerin, there is some degree of instability, especially when you add pollen extracts. We found that cat allergen, FeLD1, is highly resistant to even cockroach extracts. So, depending on the allergen, you're going to get varying degrees of resistance to insect proteases. But, if you really wanted to apply that, you'd have to remember every combination and say, sometimes you'd be able to mix and sometimes you can't. So we recommend that if, instead of having technicians trained on every combination, that you avoid using cockroach extracts regardless of glycerin content in terms of mixing with other allergens.
So cockroach by itself?
Cockroach should be by itself. Now the fungal extracts, which have relatively, they do have protease activity, but we find that the majority of other extracts can be formulated with fungal extract as long as you keep it in 50% glycerin. But you'll find that most allergists do not inject, they make the dilutions in HSA or aqueous buffers. Very few allergists dilute the stock concentrate in 50% glycerin because of the pain that it induces upon injection.
It's an irritant, isn't it?
That's right. And so the compromise we've been suggesting is using 10% glycerol saline as a diluent to maximize stability, but 6 months to a year is probably the maximum stability that you would get in those fungal mite or fungal pollen combinations. So if you remember to short date those dilutions made in 10% glycerin, and you maintain the stock concentrate in 50% glycerin, you might be able to get by with many of the fungal combinations.
Yeah, I remember a long time ago we talked about this because you were one of the people who pointed out the protease activity phenomenon, and even though the allergen extract as measured by IgE binding decreases over time when protease-containing extracts are mixed with others, the question is, does that reduce the effectiveness of immunotherapy? The T-cell peptides might still be present, and maybe it's a more effective extract.
Exactly.
Has any further work been done on that?
We haven't looked at T-cell responses, but we've looked at immunogenicity from a IgG antibody responses in mice, yeah. And we do find a reduction of IgG antibody responses when extracts are highly degraded by proteases.
So maybe it is affecting them. Brock Williams has a question as always. So put it away, Brock.
Hey Bob, how you doing?
Hey Brock, pretty good.
Good. Oh, I got lots of questions here, but I think the key is has anyone ever determined the specificity of a skin test with any allergen extract?
Specificity meaning
you get what you think you're getting. Yeah.
To IgE specificity?
Well, no, the skin test itself with an extract. I mean, I don't think you can do it, actually. It's kind of a trick question. Mm-hmm. And it relates back to the sensitivity. And they say, well, it's very sensitive to put these things on the skin, but they never say anything about specificity, and there's a good reason. I just want to bring that up.
Oh, I see what you're saying, yeah.
Yeah.
The only experience that we've had on that is using the skin test for selecting patients to do our clinical immunotherapy trials.
Uh-huh.
So it's a pretty high level. We find you need a fairly large skin test to predict symptom scores during the pollen season.
Right, and so with up to a 20-fold variation in like the grasses, what's a reasonable cutoff? I mean, is 3 millimeters a reasonable cutoff, or should it be much higher?
Someone needs to do the clinical study.
Yeah, it's frustrating not to see these things. And the only other question is, like with the dust mite, we know that about 20% of the people are not sensitized to DRP 1 and 2. There, 20-some allergens there, and I kind of wonder why we don't do more with components in skin testing. And I mean, like all the weeds cross-react to some extent, and most of the pollens do too, with some allergens that may not cause disease, but they can cause positive skin tests. So are you guys ever going to look at component content of allergen extracts?
Well, we do some of that in terms of quality control, but not from a clinical diagnostic approach, like the FADIA's,
Yeah.
Specific allergen IgE testing. So we haven't looked at it from a diagnostic point of view, only from a consistency manufacturing point of view.
Well, certainly a lot more to learn about allergen extracts, but we're going to have to stop there. So thanks. Brock Williams, as always, has the final word. We'd like to thank Bob for this presentation. It was really illuminating, as always. We look forward to finding out more about the standardization of cockroach. We're very interested. We're actually finishing up our practice parameter on cockroach right now. Maybe you could take a look at that and see if we're in the right track. At any rate, so thanks again, Bob, for this presentation. Just remind everybody that next Monday we're going to be dealing with difficult patients, and we're also going to be talking about food allergies. So join us again on Monday. In the meantime, have a great weekend, everyone, and we'll see you next time. Thank you.
Thank you, Jim.
This has been an ACAAI production. To learn more about Conferences on Lyme Allergy or the American College of Allergy, Asthma, and Immunology, go to acaai.org. See you next time.